Hi class. So let's talk about pH homeostasis. So this is another function of the renal system, and our normal body pH ranges between 7.35 to 7.45. If it goes below 7.35, we have metabolic acidosis. If it goes above 7.45, we have metabolic alkalosis. Now, there are three ways to regulate pH. First, there is a blood buffer system. This is the quickest way, but this system kind of gets overwhelmed very easily. Then we have the respiratory compensation mechanisms, which is little slower but more steady, I would say. And the best way to regulate this is our renal system. It's the slowest but the most effective way of regulating our blood pH. So this is the equation. We have talked about this before. If you have more carbon dioxide, that combines with water to form more carbonic acid. And if you have more carbonic acid, the equation shifts to the right, meaning you will have more hydrogen ions, and vice versa. So this is our blood buffer system, and this system resists change in pH. Now, I want you to know that the bicarbonate ions, they are the key players that resist pH change. Again, remember, the blood buffer system is very, very fast, but again, it gets overwhelmed super easily. Looking at the respiratory compensation mechanisms, if the pH is, if it gets too acidic or too alkaline, 75% of the problems are taken care by the respiratory system. This is a quick process and also quite efficient. When we hypoventilate, that means we are trying to conserve carbon dioxide. We are keeping carbon dioxide in the body. And when we keep carbon dioxide in the body, that means we will generate more hydrogen ion. And that means that for some reason our body is alkaline. That's why to balance it out, we are keeping in carbon dioxide, or we are retaining carbon dioxide. When we hyperventilate, we are breathing out more carbon dioxide, right? If we breathe out carbon dioxide, of course, the concentration of hydrogen ions will decrease, and this happens when typically we have our blood pH tends to be a little more acidic. So in that case, we just try to get rid of the carbon dioxide and we hyperventilate in that case. All right, the renal compensation mechanisms. This is the most effective way to maintain the pH balance. And it takes place in the PCT, DCT, and also in the collecting duct. This is as I mentioned before, this is longest but most efficient process. It takes sometimes, hours to maybe a couple of days. So let's look at how the pH is regulated by the proximal convoluted tubule. In light green, you see a transporter in the luminal side. So I have highlighted the lumen in blue, right? So in the lumen, we have the filtrate. So we have filtrate in the lumen. And then this is a blood capillary, and in between, this is interstitial space. Good. Okay. So we see the lumen, we see the blood, we see the cells of the proximal convoluted tubule as shown in black. We have interstitial space, and we also see the blood capillary. Okay, so there is this light green color transporter on the luminal side that pumps out hydrogen in exchange for sodium. So sodium comes into the cell, hydrogen goes out. The hydrogen that goes out into the lumen combines with bicarbonate ions to form carbonic acid, as you see here. And then this carbonic acid dissociates to form carbon dioxide and water. Now we have this carbon dioxide and water in the filtrate, right, in the lumen. They diffuse into the cell, as shown by the big red arrows. They diffuse into the cell, and within the cell, you have this enzyme called carbonic anhydrase. Carbonic anhydrase helps carbon dioxide and water to combine to form carbonic acid, and then carbonic acid dissociates to form hydrogen ion and bicarbonate ion. And sorry, and bicarbonate and sodium moves out of the cell into the capillary along their concentration gradient. So this is diffusion. These two come out by diffusion. Bicarbonate and sodium come out of the cell by diffusion. This is how the proximal convoluted tubule reabsorb bicarbonate. So what are we seeing? That bicarbonate is being absorbed or reabsorbed back into the blood capillary. And why is that happening? Because first, we don't want to lose carbon, we want to maintain our blood buffer system. And most importantly, this happens if our system tends to get a little acidic. Of course, we will hold on to the bicarbonate. All right. pH regulation by the distal convoluted tubule and the collecting duct. Easy thing. Now, there are specific cells called intercalated cells that are located in the walls of the distal convoluted tubule and collecting duct, and they respond to changes in pH. There are two types of intercalated cells. We have type A and type B. Please remember that type A regulates or reacts to acidosis, type B reacts to alkalosis. And typically these two types of cells, they regulate hydrogen ion, bicarbonate ion, potassium and chloride ion concentrations. Type A and type B are exactly similar. They are exactly similar. The only difference between these two cells are the locations of the transporters. So they have the same transporters, but the location of the transporters are different. And that leads to some basic differences in type A and type B cells, which includes that type A cells increase potassium concentration in blood and type B cells decrease potassium concentration in blood. So let's take a look now. So here we are looking at pH regulation by type A cells. Type A cells, remember, they respond to acidosis. So there is a specific transporter located on the basolateral side, which is shown in green here, and that transports bicarbonate out of the type A cell into the blood vessel in exchange for chloride. So chloride comes in, bicarbonate goes into the blood cell. Another transporter shown in yellow, it pumps out hydrogen into the lumen in exchange for potassium. So hydrogen is pumped into the lumen, meaning hydrogen will go to the filtrate and be pumped out of, and will be, we will just get rid of the hydrogen ions. So this happens again. When do we need to get rid of hydrogen ions? When we are acidic, right? We need to get rid of hydrogen ions, and that is exactly what's being controlled by these type A cells. Okay. And then as hydrogen is pumped out into the lumen, potassium ions are pumped in, and then ultimately potassium leaks out into the blood vessels. Therefore, these cells are helping to reabsorb bicarbonate and excrete out hydrogen ions, and in the process, a lot of potassium are reabsorbed back into the system. Moving on to pH regulation by type B cells. Again, they are exactly the same, just the location of the transporters are different. As you can see, the green transporter is now on the luminal side, and it's doing the same thing. It's just pumping out bicarbonate, taking in chloride, but instead of pumping it into the blood vessel, it's pumping it into the lumen. So essentially, we are losing bicarbonate, right? And when do we need to lose bicarbonate? When we get a little more basic. And the yellow transporter, that is also present, but again, see, it's on the opposite side. So hydrogen is pumped back into the blood vessel in return for potassium. So in this case, we are retaining hydrogen and we are getting rid of bicarbonate, right? And again, remember, we are probably a little basic at this point. So that's why we are holding on to the hydrogen ions. So all the potassium that comes into the type A cells, they leak out into the lumen. So in this process, we lose a lot of potassium ions. So that's it about pH regulation, and that's all about my lecture on the renal system. And thank you.